Hi, welcome back. I'm Dr. Carl Goldkamp. Most people think they have no control over their aging, and that's so not true. There's a number of reasons that for the last 70 years, we've been accelerating our rate of aging, unlike all previous generations. Today, I want to show you why that's the case and how you can change that, starting with nutrition. This is the big picture look, remember. We're going to look into dementia and Alzheimer's, but also cerebral vascular and stroke, cardiovascular, heart disease, immune relative to autoimmune disorders and cancer. We've mentioned that already a number of times. Metabolically and diabetes and obesity. It's not just on these particular deficiencies alone dramatically affect these. So we're not looking at all deficiencies. We're looking at just these three. And we're looking at food sources of these. Okay, here's one that not too many people talk about. It says, not to mention the incidence of autism has increased enormously. This is since '87, and there's a number of graphs that I could have chosen to show you how quickly. This only went up to 2007. If I was to continue it to up to 2022, it's 1 in 54. But I wanted to show you the trend is outrageously high. You could say other things have increased too, like epilepsy and mental retardation and cerebral palsy, but not to the extent of autism. How did this happen? Certain foods that past generations were required to have were not consumed very often by this generation. What foods are they? Liver, egg yolks, fish liver, or fish liver oil. So there was generations that hated having to grow up with that tablespoon of cod liver oil every day. It served them well. There was generations of people that grew up saying you have to clean your plate. You had to eat your vegetables and meats, but they had liver more than a few times a week. They had to have that. They grew up having it. Now, most people don't like it. They don't have it. They certainly don't have fish liver oil. Egg yolks, we have been talked out of having eggs over the last 70 years because we were afraid of the cholesterol that egg yolks would give us. That was completely bogus information that we've talked about in former videos. But consequently, we don't have eggs and we don't get the other nutrients that are in those egg yolks. And also, you can say that the chickens are raised in a way that the egg yolks are not as nutritious as they were 70 to 100 years ago. All true. And then liver in general. Okay, so the underlying commonality with these conditions are really just two things. They have the same collective nutrient deficiencies because they don't eat these particular foods, and there's a subpopulation that are genetically predisposed from the mutations that they have that slow down their whole methylation process. We're going to talk about it a little bit. So it is true that with good nutrition, it fixes a lot of conditions. You give good, valid nutrition to pretty much any condition, and there are some genetic exceptions to this, but for the most part, we're talking about 99.9% of all conditions. Give them good nutrition by good whole food. So good egg yolks, good liver, good whatever they're having, you make it real food and remove the environmental toxins, or give them the foods that do not have those contaminants, and they will do real well. However, it is also true as nutrition becomes poor, that is, the nutrients become lower and lower and lower on a daily basis because they're eating the wrong kinds of foods or no longer eating the nutritious foods, that population with these mutations are much more vulnerable. Good nutrition raises all boats like a rising tide. Bad nutrition makes the vulnerable much more vulnerable. And those are the first people that suffer all the illnesses that we've been talking about. So poor nutrition, nutritional deficiencies, make some people far more vulnerable than others. Specifically, those with a much greater need for these specific nutrients due to their methylation SNPs, their single nuclear polymorphisms. Okay. Who's most at risk with those specific polymorphisms that affect their ability to maintain a healthy methylation pattern? Let's give an example. Here's dementia. We're not going to go too deep into these conditions. We're going to just look at this one as an example. We can estimate the possible impact of the modification factors. In other words, Alzheimer's or dementia is a treatable condition that can improve. You're not stamped with a genetic predisposition. You may have a predisposition, but like I said, you can give good nutrition and it raises all boats. You give good nutrition. So here's what the research shows. That population attributable risk, PAR, high homocysteine levels, or in other words, low B vitamins, 22% of all cases, about a quarter of all cases of dementia are due to low B vitamins slash polymorphisms that require higher amounts of B vitamins and low fish intake. Here is the thing. If you just give B vitamins and these individuals still have low omega-3, they won't get any increase. You will not have diminished the high homocysteine. If you give them omega-3s but don't give them the increase of B vitamins, that does not change their risk. These two have to be given together. The fish oil, the vitamin, the omega-3, the DHA, and the EPA have to increase along with the B vitamins, specifically the folate, the B12, and the B6. Here's an example. Dementia, including Alzheimer's disease, can be prevented. And this is a statement, an international statement saying that the G8 Dementia Summit, signed by 111 scientists from 36 countries, basically saying that you can reduce the risk by at least 20%, if not a quarter. Omega-3 fatty acid treatment reduced mild to moderate Alzheimer's diseases. No benefit effect on cognition of supplementation with omega-3, apparently failed trial, but in just those patients with low homocysteine and added the omega-3, they benefited. So omega-3 fatty acid needs to interact with B vitamins in order to benefit. You need both of them together. So don't be thinking, I'm going to take my omega-3s, I'm going to be smarter or have lower risks of Alzheimer's. No, it's a little more holistic than that, okay? Makes sense. Conclusion from this particular study is that omega-3 fatty acids appear to protect the brain in people with low homocysteine with good B vitamin status. So we'll measure that. We'll look at that in a second. B vitamins only appear to protect the brain in people with good omega-3, just the opposite. So you add B vitamins and get that low homocysteine, but if their omega-3s are not adequate, it doesn't matter. So these unexpected interactions could explain why some omega-3 trials have failed and why some B vitamin trials have failed, because they were too independent. They weren't locked in with each other. This is really interesting. So this is the regions of the brain that are affected by B vitamins. So your whole brain isn't affected by B vitamins, just these specific areas. And if you notice, there's an area down here called the amygdala, and that's where your memory is. And so when you're behind on B vitamins, it's going to be disproportionately affected. So your memory and the thing that the amygdala is connected with are going to be affected. Okay, B12 within the normal range. This is really interesting. This is all the normal range. This is really teasing it out, but they're saying those who have a high normal range have far less. This is a 2-year measured period. Within the course of 2 years, those who had high normal levels within normal range had far less brain atrophy. Those who had low normal range had far greater atrophy. That is a big deal. Okay, here's one of the many Norwegian studies, cognition in the elderly, intake of fish. What does fish have? It has the omega-3, it has the B vitamins, it comes in a package. How convenient, right? Okay, so over 2,000 Norwegian elderly, it shows gram of fish per day, and look at the difference. Absolutely. The more fish, the better they did on their memory score. That's 2007. So this is interesting. So this again is about B vitamins and looking at, this is before and after 2 years. So no B vitamins, big atrophy in 2 years. This is the bottom of the brain looking up. This is where your amygdala is. And this is no B vitamins, and this is with B vitamins for 2 years. Hardly any change, very little change, big change here. So that's just on B vitamins, those parts of the brains that are affected by B vitamins, which is memory, among others. Yes, so here's we got the methionine cycle. It's all about B vitamins. Here you go, folate, B12, B6. 3 B vitamins regulate the level of homocysteine. We're going to measure that and see what that's measured. And here's your glutathione. You've seen this in other contexts. This is the methionine cycle. Some people call this the methylation cycle because it kicks off these methyl groups. CH3 is a methyl group. It gets kicked off to go over to hook up with to turn a gene on or off. And there's SAM, accidental methionine, accidental homocysteine. Okay, what about it? Here's a bigger picture. Here's the methionine cycle. Now here's some of those genetic polymorphisms that people have. This is just 3 I'm picking out. This is one that most people have already heard about, MTHFR. I've mentioned it a couple times. It's not just that, it's the context of. But here we go. We're looking at the same thing. Methionine cycle, folic acid cycle. So here's your B12. Here's your B6. Here's your folic acid. Boom, boom, boom. These are really important. 3 levels we're going to look at. Impairment of any of these 3 enzymes will create a potential major health problem. So I'm saying, we talked good nutrition raises all boats, right? Everybody gets better. Bad nutrition disproportionately hurts those that are most vulnerable. Those who have these mutations, these polymorphisms, are disproportionately more vulnerable to a lot of the problems we've been talking about. Okay, low vitamin D is a risk factor for Alzheimer's disease. Look at that. Severely deficient and conversion to dementia. That means they qualify, they become demented. Okay, severely deficient, became demented. Adequate, not many. So it's pretty straightforward. These are the number of years. Within 8 years, being consistently low in vitamin D, your chances are pretty high you're going to be have dementia. And that was from 2014. That is 8 years ago. So we got to talk about homocysteine. You ready to talk about homocysteine? Okay. So here we go. Here is something I hope your doctor is looking at. This is just me taking a spreadsheet of some of the people we've seen, making their names obviously anonymous. But what I did, I lined it up just relative to their homocysteine level, right? High homocysteine versus high normal, right? So this is definitely high, and the purple is high normal. And I just didn't do all labs because that would be too much to look at right now. But I wanted to show you these are the most common out-of-range results. Homocysteine, folate is red blood cells, methylmalonic acid, B12, vitamin D, inflammatory markers, and the omega-6 ratio. Look at this. Look at the correlation between elevated homocysteine. Look at all the red. And now go down and look at the omega-6, 3. They're almost mirroring each other. Almost mirroring each other with some very similar numbers as well. That's amazing. So what I'm saying is this is indicative. So this person here obviously has either, right, greater need for the B vitamins that we've talked about because they have these mutations, or they are not getting, or they have really terrible nutrition. They're not getting the B6, B12 folate. And apparently, they're getting too much vitamin 6, omega-6 versus omega-3. These together, the chances for these people becoming demented earlier and earlier, aging faster than they have to be, accelerated aging, is very high. Those that are lower, as we're going that way, isn't it amazing? These go neck and neck. Inflammation, highest here, lowest down that way. What else can we? And it goes on and on. They did glucose, lipids, so on and so forth. But I think this box really explains it all. And this is what we've been talking about. Then when you add in vitamin D deficiency, look at that. This person happens to be a guy, 46-year-old guy. He was high homocysteine, low vitamin D, high omega-6 versus 3. Bang, bang, bang. And so now what we're going to do is look at what we call intercellular. This is the bottom part of that. So now we're looking within the white blood cells to find out how much of these nutrients, these micronutrients, are there. What we're looking for is functional deficiencies. So this is B12, same person. Yeah, kept the line up the same. Look at this. This is intercellular. These are things we can address, and we did address, and they do make a change. So there you go. Definitely deficient, and then borderline deficient was folate. And you can add in a few others. Here's the next person. Folate. Oleic acid, you can get that from olive oil and so on. So you get my point. Here again, we're looking at methionine cycle, folate acid cycle. This is called neurotransmitter. So there's tryptophan to serotonin and dopamine. You can see how all of these are affected. Glutathione is down here. Here is homocysteine. Homocysteine becomes high because you're deficient in B vitamins or you have a greater need. The greater need speaks to the genetic polymorphisms that you have, otherwise known as SNPs. So that's a blood serum level that we can measure. There's your SAM. There's B9 is folic acid. This is simply I like the way they do it. It's a simple, these are all cofactors, and these are some of the enzymes, right? So this is, over here would be the MTHFR. This says this person is deficient in folate, they're borderline deficient in B12, there's your B6, they appear to be fine. And so it puts it into place by saying, we clearly can help this person by getting their nutrient levels up higher. All right, the 3 genomic bottlenecks I mentioned before. CBS, called cystathione beta synthase, which is basically going down to make glutathione, which should be over there. MTRR, that's another link between folate and B12, and then MTHFR. Boom, boom, boom. And if people have all 3 of those, so here is an example of all 3 of those. Here's a person he has the worst mutation here. He has the worst mutation there, and he has heterozygous, right? So that's homozygous, homozygous. Homozygous is more than one problematic gene. So this person's definitely going to have a greater need for those nutrients than somebody else because of their genetic predisposition. Okay, again, this person looks like that. Blocked, blocked, blocked. Not completely, otherwise they'd be dead. But they need more. They need a higher amount. Just like we saw that graph between normal B12, high normal, they're doing fine. Low normal, there's a big difference. Okay, let's look at MTHFR, the SNP. Here's me and Judy. We're both homozygous. There's a different person, just for MTHFR. These are the 2 genes. This is the worst to have. This person's heterozygous here and heterozygous there. So they're a bit impaired, not that much. And this is the least impaired, right? Heterozygous for just the least offending gene. Okay, Carl SNP's family history of alcoholism and dyslexia. That's me, homozygous. That's a problem for the worst gene. I'm partially impaired by these other 2, but I'm heterozygous. And I'm also partially impaired because of my conversion to glutathione. That's a setup for a lot of problems. It means I have a problem making glutathione. I have a problem activating my folate to methylfolate and methyl B12. Here's somebody else who's insulin resistant. Look at there. Homozygous for the worst gene. Heterozygous for both of those. Not so bad down here, but that is a problem. Here's a person with history of heart problem. MTHFR, they're heterozygous for both. They're homozygous for one here completely, and not much down there. We have these 5 nutrient deficiencies caused the incidence of chronic diseases to skyrocket. So these 5 deficiencies we've talked about, they are the omega-6, the vitamin D, the omega-3, the vitamin D, the B6, the B12, and the folate. But there's primarily just 2 reasons these have existed. The first reason is people's diets have severely changed in the last 50 years. You can say, yes, it's about processed food. That's correct, but it's not the whole thing. We kind of stuff a lot of things into the whole processed food argument. Clearly, it's lack of nutrition and it's high in omega-6. We got that, covered that in at least 10 other videos. But it's more than that. Most people don't consume as much liver as previous generations by a lot. Most people don't eat eggs in the quantity as previous generations did due to the fear of increasing their cholesterol. The quality of grocery store eggs is far inferior to pasture chicken eggs. The yolks of grocery store eggs have about a 6 to 8 omega-6 to 3 ratio, whereas before it was 1 to 1. So go back 100 years ago, it's about a 1 to 1. You're getting pasteurized eggs from chickens that just allowed to walk around the yard. Most people don't consume as much fish as previous generations did. They didn't grow up taking cod liver oil, omega-3, vitamins D and A. Even the Catholic Church stopped requiring those who are that religion. They didn't have to eat fish on Fridays anymore. Processed food consumption is low nutrient food, high omega-6 oils. So the second reason is these combined nutrient deficiencies affect those with the Wirth methylation SNPs, far worse than those that don't have such mutations. All right, getting what we need. Here's basically it. Chicken's diet should be, chickens are not meant to be vegetarian. Despite what you find in the grocery store stamped on your carton, chickens have natural diet of worms, beetle, grasshoppers, mealworms, as well as plant scraps. Eggs from chickens with access to green plants, insects look a lot different than eggs from factory-raised chickens that never go outdoors. There you go, free-range pasteurized, almost orange. Caged, that's not a bad-looking yolk actually, you get them down to yellow. Okay, fish oil and fish liver. Think of the Inuits. They were high omega-3 and vitamin D, and they didn't really get any sun. So where'd they get it all? They ate fish livers. They ate walrus livers. They ate seal livers. So that's their hanging salmon. This is where you can get, so actually get fish liver, and these are really good. We get this, and you can get it on Amazon. And there's cod liver oil. This is basically what you have to do today if you're not a native Inuit. And probably, unfortunately, in the last 70 years, we have polluted both the poles, primarily the North Pole, and there's been a tremendous increase in environmental toxins in the fish they eat, and so that's another issue. So the remedy is dietary fish or fish liver. And yes, there's always sun and supplements. Sun will take you so far, but you really need to think about what you're eating. Okay, when was the last time you had liver? It's the best source of dietary B12, folate, and B6, all in one sources of food. The choice is always yours. Till next time. Hi, I just wanted to mention, if this is something you're interested in, finding the nutritional value of food in general, relative to deficiencies, relative to various chronic diseases that we have, you really would appreciate this following video over here and a whole series I put together about that. Not just about epigenetics, but about nutrients and about food. Something we all need to take responsibility for, our own health. Till then.